Okay, good afternoon, everyone. So everyone have joined. There are 56 participants. Your exams are over, no? Okay. In the last lab, we have done up to fifth experiment, your journal bearing test trick. Okay, so have you started writing the journal? Have you started writing? So your people have not came to college? You were telling
No,
me
ma'am.
you are going to join college?
From 21st March we are allowed, ma'am.
Okay, all of the students will come?
Some are coming, ma'am. Not everyone is coming. Some of them coming in April.
Okay, so when is your final exam?
Ma'am, April 25th.
25th April. Okay, so all the students will come by April 25th, no?
Yes, ma'am.
Okay.
Ma'am, and we did not get any reference from our seniors also. MD Lab.
Okay. तो उसकी सॉफ्ट कॉपी कैसे दूंगी मैं? I will take pictures of one or two experiments, okay, and will share it with you. So with reference to that, you can write the other practicals also. Because aim apparatus and all these things and procedure, you all know it. So I will share the screen, the pictures of the journal. Okay, I will answer about it. So I will let you know. And up to third assignment you have completed now. Yeah. So next your sixth, seventh, and eighth experiments are based on polariscope. Okay. So your sixth experiment is study of plane and circular polariscope. Seventh is study and demonstration of isoclinic and isochromatic fringes. And eighth is measurement of fringe values of birefringent material. Okay. So today we will study about sixth and seventh experiment, because in this polariscope only we will get these isoclinic and isochromatic fringes. So I will let you know about this also. Yeah. So have you studied about polariscope? In your theoretical part or in any practical? Do you have any idea about like polariscope? What is it? So up to this semester, up to this time, you have for the stress analysis, you have used the analytical method, or we can say the theoretical method. Like in strength of materials, you have evaluated the stresses using the formula based. Okay. So for stress analysis of any component, we have three methods. First is analytical or theoretical, which you have already done in strength of materials. Second is numerical analysis, which is CAE based, that is finite element analysis based. So you are studying finite element analysis, FEA, FEM. Do you have that subject right now? Finite element analysis?
No, ma'am.
Yes or no? No.
No.
Okay, so you will be having, I think, in final year. And the third is experimental method. So in this experimental method, we use this photoelastic method. Okay, photoelastic principle for the polariscope. So what is this photoelastic principle? It is an experimental technique to find the stresses in a object. Okay, so in this what happens is we have, make a setup in which we use a polarizer and an analyzer combination to see the fringe patterns. Okay, so these fringes are these fringes demonstrate the level of stresses in the object when we apply load in the object. Like in strength of materials, you have you have analyzed, I think one block is there and you are applying some tensile force on it. So you have evaluated the amount of stresses producing induced in the object using formulae. So here what we do is we have an experimental setup that I will show you, and its setup is also there in our college. We have in last lab, I think I have only taken it. I will share the screen with you. Hmm. Yeah, everyone is able to view it. So there are two polariscopes shown here. Actually, there are three polariscopes: plane polariscope, circular polariscope, and an elliptical polariscope. Okay, so we have used circular and plane polariscope. In plane polariscope, what happens if there is a source of light? Let me show you the video first, I think. Then you will get a clear idea. No? Uh, okay. So this is an experimental setup. This video I have taken from site of other institute, but similar type of setup we also have in our college. Okay, so this is a light source you can see here. Uh, at this back side you can see this light can be a monochromatic light or white light. Monochromatic light means only one color light will be there, like yellow color light usually we use. So only yellow color light will be there or white light will be there. Okay, so if we are using a yellow color light as the light source, then the fringe pattern we will get will be a black and white pattern. And if we are using a white light, then it will have the wavelength of all the colors. Therefore, we will see a colored fringes. This is the difference between white and uh, monochromatic light. Okay, so in this case, I think they have used white light. You will see. So this is the source, light source. Then here they have placed a polarizer. The function of polarizer is to make the light in one wave. Okay, because when the light is coming, it have wavelength in every direction. So it will depend on the orientation of the polarizer, that in which direction it is oriented. In that direction only, it will allow only that wavelength to pass through it. Just a minute. So this is a polarizer and this light is coming. So this is unpolarized light because it have a wavelength in every direction. Okay. After passing through polarizer, if polarizer orientation, you can see these vertical lines. If this is oriented vertically, it will pass only the vertical waves. If it is oriented in horizontal direction, it will pass only the longitudinal waves. Okay, or the horizontal waves. Okay, so this is the function of polarizer. समझ में आ रहा है ना सबको अभी तक? Are you all getting it? Are you able to understand it? What is polarizer? What is its function? Yes or no? समझ में आया पोलराइजर क्या है? अरे कोई लिख ही दो चैट पे यार. Or should I explain once again?
Hey.
What? Okay, you have understood. So now I will show you the. So this is the experimental setup. Okay, so here it is the light source, then a polarizer is used, then this specimen is there, which we load in the diametric manner, and then there is analyzer. Analyzer is nothing but polarizer, but with different orientation. Okay, so if the analyzer and polarizer both are in same orientation, then the light will pass from it, and the background, there will be effect only on the background and the fringe pattern. The fringes will be opposite of the opposite pattern that we will see afterwards. So we put it in such a manner that polarizer and analyzer are perpendicular to each other, and from here the viewer see the fringe pattern. From point A, you can say from this point, the viewer see the fringe pattern in the specimen, and these fringes, the more the number of fringes it demonstrates, the more stress level is there in the specimen. So in this experiment, they are performing the experiment. So this is the analyzer. This screen you can see, this is analyzer. This is the specimen. Okay. So you can already see there are fringes on that. So we are just placing it, and this is made up of, this specimen is a glass specimen. It is made up of epoxy resin. Its material is epoxy resin. Okay. So when the load is applied, you can see here a 10 kg load is applied. Then the fringes have started developing in the specimen. So this shows the stress level in the specimen. Okay. So as we increase the load, more number of fringes will be there. So here we can see that they are increasing the load. These are the colored fringes. It means they are using white light. You can see the color green, red, and here there is some blue. These are the fringes. Okay. This is showing the stress level in the specimen. So what happens is when we apply load, the first fringe appears here, and the fringe from both the sides, from upper side and lower side, merge at the center, and then it moves towards the side of the specimen. Okay, so this is the 0th fringe, we say. Then now you can see here, these fringes are merging because they are applying the load, they are increasing the load. and the outside fringes moving towards the outside of the outside border of the specimen. Okay, so as we increase the load, this fringe also will merge and it will also move outside. Okay, so finally we will get, you understood it, no? This is showing the stress level in the specimen. Initially they have applied a 10 kg load, but after that they have increased the load, you can see here, and a more number of fringes are we can observe in the specimen. It means the stress level is increasing in the specimen. Okay. And these are the isochromatic fringes. Isochromatic means fringes of constant color. And in these fringes, the difference between the principal stresses remains constant. That is, along this complete contour of the fringe 1 or other fringes to be appear, the difference between the principal stresses will remain constant. So therefore, there are these are isochromatic fringes. Okay. And so this was a plain polariscope. Here there was not no retardation of the wavelength. Okay. In circular polariscope, we provide a retardation. So here we have two fringes are there. I don't know, show नहीं हो रहा है clear. Here you can see two types of fringes are developed. These are the isochromatic fringes, and this is the fringe number we give. Like this was the 0th fringe, then it will again combine and merge and then move outside. Then it will be first fringe, then this will be second fringe. As we keep on increasing the load, these fringes will start developing, and it will the number of fringes will be increasing as you increase the load. Okay. So the more number of fringes are there, the more stress level in the component is there. And these are isoclinic fringes. So in this video it was not clearly seen, but actually in plain polariscope we see both a combination of isochromatic as well as isoclinic fringes. So all the fringes don't like appear at once. These are the result of different experiments they have performed by rotating the analyzer and polarizer combination by 10 degree, 20 degree, 30 degree, 40 degree, 50, 60, 70, 80. Okay. So at one time we get only one fringe. Suppose we have this fringe, so we have a combination of this and this opposite fringes will be there. So these just two fringes appear along with these fringes. Okay, it will be clear in other video I will show you. So these are the isochromatic and isoclinic fringes. So there are two types of fringes appear. In isochromatic, what we have seen is the difference between the principal stresses, that is sigma 1 and sigma 2 remains constant for one fringe. For 0th fringe, it will be some value. For first fringe, some value will be there, but the difference will be constant. While for clinic fringes, the values of sigma 1 and sigma 2 may be different, but its direction will be constant along the fringe. Okay. This is the two types of fringes we get. You understood it, no? Both types of fringes, isochromatic and isoclinic. So this definition you have to write in your manual, okay? In the theory part, you have to write what is a polarizer, what is analyzer, and what are the types of fringes we get and its definition. So you must have a clear idea about it. And this was actually your seventh experiment, the study of isoclinic and isochromatic fringes, but it should be taught in combination only, because here you are seeing the fringes and you don't know what is it, then it will be not beneficial for you, right? But in experiment, there are two experiments. The first is only the setup, and in second experiment you have the study of fringes. Okay. And till now we have studied about the plane polariscope. Now second is circular polariscope. Okay. So in circular polariscope we have this type of setup. There is a this is same, the source is there, a polarizer is there, analyzer is there, but the difference is we have two quarter plates here, first quarter wave plate and second quarter wave plate. The purpose of first quarter wave plate is to retard the motion of the wave by an angle of 90 degree. Okay, so there are two axes, fast and slow axis, and here we have the opposite. The axes are changed for this second quarter wave plate. This is in place of slow axis, it is fast axis, and in place of fast axis, it is slow axis here. Okay, so the only purpose of this is first quarter wave plate will retard it. So from the source to the polarizer, light is coming, then from polarizer to first quarter wave plate, the plane light is coming, plane wavelength light is coming, then one wavelength light, and after passing through quarter wave plate, it will be a circular wave. Okay, so circular wave will be entering the specimen. Here what was happening, the plane wave only enters the specimen. Here the circular wave, that is a retarded by 90 degree wave, enters the specimen. So in this case we don't see the isoclinic lines. The lines I have shown you here. No? These lines are not, these fringes, isoclinic fringes are not appeared in the circular polariscope. So if in case of plane polariscope, we have both the fringes. In circular polariscope, we only have the isochromatic fringes. We don't see the isoclinic fringes. And this is an advantage because while studying the fringe patterns, while counting the fringes, sometimes it becomes difficult to count it, because both the fringes gets overlapped at some places. Okay, so in circular polariscope, it is easier to count the isochromatic fringes. Okay. So the final will be a plain light only, because this first quarter plate have retarded it. Then when it passes through the second quarter plate, it have um, uh, again it have actually accelerated it, because it have neglected the effect of first quarter wave plate. Therefore, it is in opposite direction. Okay. So finally, we will get a plane wave only, which was passing through the polarizer from here to first quarter wave plate. Okay, so this is the difference, and I will show you one more video I have ready to start with. Hmm. यहाँ तक समझ में आया ना सबको अभी? Have you understood difference between plane and circular polariscope? I think we have not used circular in last time because the quarter wave plate was not available, I think, in lab. We have done only this plane polariscope only. And there is one video. And in this video you can see it. Okay. So these are some of the lectures by NPTEL. So IIT professors take these lectures. So in this they have shown it very clearly. This is a plane polariscope. This is the source, polarizer, specimen and analyzer. So they have shown how we rotate the analyzer and polarizer combination. Okay, so these are the these are the isochromatic fringes, and these curved lines you can see, these are the isoclinic fringes. Okay, so these it is a combination we always get in plane polarizer polariscope. Okay, so as we rotate the combination of polarizer and analyzer, the isochromatic fringes remains constant because it is just showing the level of the stress. But isoclinic fringes shows the direction of the principal and both the principal stresses. So as we change the polarizer and analyzer, the isoclinic fringes will rotate throughout the specimen. Okay, and this is for circular. This is a circular polariscope. Here we have polarizer, then first quarter wave plate, a specimen, second quarter wave plate, and analyzer. So the only difference when we keep it parallel, the background will be white because the light will pass as it is. Light don't have any blockage, okay, obstruction. But when analyzer is perpendicular to polarizer, then the background will become dark and the white fringes will become black and black will become white. So you can see it here. Here you can see. Just observe the difference between these two. In this case, the polarizer and analyzer are parallel to each other. The background is white. You can see it. Okay. And these are the fringes. This we count as zeroth. So zeroth fringe. Then this will become, when it will combine and move here, it will become first fringe, and so on. The number of fringes will increase. And when we have rotate the analyzer perpendicular to the polarizer, then the background will become black and the fringes have changed. Here it was this was the zeroth fringe, and here now this is the zeroth fringe. Okay, so this is the change in pattern of fringe. So this is the difference for plane, for parallel and perpendicular analyzer polarizer combination. And I think they have also these are the various setups we use for plane and circular polariscope, the orientation of polarizer and analyzer, the quarter wave plates, and the background we get. Okay, so you can either take a screenshot of it because you have to make this table in your manual. Okay, different types of setup we can have for both polyscopes. So take a screenshot of it and put it in your journal. You have to make it. Yeah, so one thing you have observed here, we are getting black and white fringes, okay? So what is the type of light we have used here? Monochromatic or white light? Can anyone tell me? In earlier case, I have shown you a video there, we have seen colored fringes. So what light was used there and what light is used here? Monochromatic here, no? In this case. Okay. And in earlier case, we have used the white light. So it has shown the colors. Okay, circular. And this is some theory part. So we don't need to go into that depth. So you have taken the screenshot of it now. So here you can clearly see this is the light source, this is polarizer, and here you can see there is a gear type of combination is there. So using this we rotate the polarizer and analyzer. So initially it is like if it is vertical, it is parallel, then we can rotate the analyzer to the 90 by 90 degree by rotating this. Okay, this mechanism we use to rotate it, this circular plate, this is analyzer. And here we keep the specimen. Their setup is somewhat different, but our setup is similar to this only. Here you can clearly see. Yeah, so here they have used monochromatic light, so we are getting some black and white fringes here. They are applying load. I think load value is displayed here. This 34.1, I think you can see here. No? And this is the angles written. You can see. So using this angle, we can, here also there are angles written, and here also on analyzer also angles are written. So using these angles, we can make it perpendicular or parallel to each other. So in this case, can you see the fringe pattern and tell me whether the polarizer and analyzer are perpendicular or parallel? Here the polarizer and analyzer are perpendicular or parallel. Seeing this fringe pattern. Can anyone conclude? Parallel होता है तो क्या होता है? Background will be white. Okay, because the light passes as it is. There is no obstruction. So we can see that it is perpendicular because the background of the specimen we can see here is black. Okay. मतलब लाइट सोर्स जहां से आ रहा है ना, तो वहां से लाइट जो है वो उसको ऑब्स्ट्रक्शन हुआ है बिकॉज इट इज vertical we say and it is horizontal. Analyzer is in horizontally oriented and polarizer is vertically oriented. Therefore, we have a dark background. Okay. So these things you have to observe very clearly. Okay. And they have rotated it. They are rotating it. So you can see here the isochromatic lines are same, but isoclinic lines are changing. Observe here carefully. You can see the isoclinic lines as we are changing the orientation of the analyzer. Okay. The load applied is constant. In this case, the load applied is same. We are not changing the load. Therefore, the isochromatic fringes are not changing, because isochromatic fringes shows the value of the stress level in the specimen. These are not changing. Okay, but clinic lines are changing. You can see here. These are changing. Okay. And this also you have to write down. You can take a screenshot of it, that if we are using white light, then we are having colored fringes, then there is a transition between colors, and then only we can say that it is a fringe. Like the zeroth fringe we have at black. Okay, so this is the zeroth fringe. Then from dull red to blue, there is a change. So it will be first fringe. This is the fringe order. Okay, so first fringe will be from dull red to blue, this transition. Then rose red to green, this will be second fringe. This is the fringe numbers. Okay, and third will be orange red to green. It will be the third fringe. So there will be, it is not restricted to only 3 numbers. There will be more number of fringes, but for a bending moment of 3, we have seen it. Okay, so as we increase the load, number of fringes will also increase. So this table, take a screenshot of it. Take a screenshot of it. This table also you have to write in the your journal. Here you have a very clear view of it. You can see the when the bending moment was when the bending was less, then number of fringes was less. Okay, there is only one fringe. Here it is 1, and here it was 3 was there. Bending moment was 3. Here, when 2 is there, then 2 fringes are there. So you can see from red to green transition, it is fringe 1, then this is fringe 2, the zeroth is black. These are the colors of fringes. So here you can see there are two fields. This is the dark field when the polarizer and analyzer are perpendicular to each other. Okay, so these are the dark fringes. So here the numbering is done like 0, then 1, then 2. Okay, so zeroth is this, zeroth fringe, then first fringe will be this, and the second fringe will develop by combination of these two fringes. Okay, so this is the numbering for the dark field, and for bright field we have it like 0.5. This is the 0.5 fringe. Okay, so then this is 1.5, this is 2.5. You can take a screenshot of this also. Here you can see that in this is for a plane polariscope, and these both are in circular polariscope. Okay, so here we can see only the isochromatic lines. Here we can see only the isoclinic lines for plane, and this is for circular. You can take a screenshot of it and try to understand it. Anyone have any doubt in this? Have everyone understood it? कोई डाउट है किसी को? try to make these figures also, no? Using some shade of pencil or something you can make, but in the theory part you have to write what is the plane polariscope, what are its what is its apparatus, okay, then how the fringe pattern we are getting in plane polariscope, and then for circular also you have to write that we are using the quarter wave plates we are using, and we are getting only the isochromatic lines or isochromatic fringes. Okay, in this way you have to write down the theory as well as make this figures also, and these both figures also you have to make in dark field and in bright field and explain it. Okay, you have to explain it in the journal. Okay, so this was about the plane and circular polariscope and the isoclinic and isochromatic fringes. So in next class we will study about the measurement of fringe value of birefringent material using photoelastic setup. Birefringent material is this only, the material we have used in these all cases. Okay. So anyone is having any doubt? Is it clear to all? We will try to perform this experiment. It is very interesting experiment. When you all will come in college, no? Once you will try to have this experiment. So this is the second type of analysis you have studied. First was the theoretical you have already studied. This is the experimental, and third type, the numerical analysis you will be studying in your final year. In that we do computer-based simulations and see these patterns. समझ में आ गया ना सबको? हो गया ना? अटेंडेंस दे दूं? कोई डाउट तो नहीं किसी को? Today is 16. Roll number 1.
Present, ma'am.
2.
Present, ma'am.
T.
Present, ma'am.
4. 4. 5. 5. 6.
Present, ma'am.
7.
Present, ma'am.
9.
Present, ma'am.
10.
Present, ma'am.
11.
Present, ma'am.
13.
Present, ma'am.
14.
Present, ma'am.
15.
Present, ma'am.
16.
Present, ma'am.
17.
Present, ma'am.
18.
Present, ma'am.
19.
Present, ma'am.
20.
Present, ma'am.
22.
Present, ma'am.
23. 23. 24.
Present, ma'am.
25. 25. 26.
Present, ma'am.
27.
Present, ma'am.
28.
Present, ma'am.
29. 30,
Present, ma'am.
31,
Present.
32,
Present, ma'am.
33,
Present, ma'am.
34,
Present, ma'am.
35, 35, 36,
Present, ma'am.
37,
Present, ma'am.
38,
Present, ma'am.
39,
Present, ma'am.
40,
Present, ma'am.
41,
Present, ma'am.
42, 42, 43,
Present, ma'am.
44,
Present.